Good morning all of you. Good morning. I welcome you all for the last day of this 5 days GIN program. Hope you have enjoyed other sessions and interact with the speakers and learn different things. So that is most important part of this program. We have conducted the program for the 5 long days so that all aspects of medicinal chemistry for the drug discovery, drug development will be covered. So I believe today's also like 2-3 more lectures are left. So some more information, some more learning will be there at the end. And after these all lectures, you'll be expert in this particular domain and can move on, can utilize this sort of understanding and discussions for your professional development, for your choosing a right directions. Okay, so with that, I'll start the lecture, the first lecture of today's. As you can see, the title of the lecture is Development of Fast Histamine H3 Receptor Antagonist for the Market. And the speaker will be again Professor Stark, and this particular histamine H3 receptor is actually developed by himself. So it's our pleasure to listen to him, and we'll start our today's first lecture. It's all about you, Professor.
Thank you very much.
Yes.
So, also once more, thank you for the introduction, and also thank you for coming here, dear colleagues. And what I would like to present today is a case study where, and I want to share with you our ideas, what we have learned, what we are following up, and in which way we really then went on. With my early introduction, I showed already where we are working on, and there were different receptors like the glutamate receptor, the NMDA receptor subtype, dopamine, the sphingosine and arachidonic pathway, but our first instant or first target was really the histamine and histamine receptor subtypes in this way. And in fact, I started my PhD study on the histamine receptor studies. At that time, we worked on agonist, and that worked out that I prepared some agonists which were quite useful. But I must say, my supervisor gave me the workload, and when we started together, he said that I have to prepare this target compound, and it took me really about 1 year to get into this target compound, and it was just some methylation derivative of histamine. So it was, in fact, it was not really a big deal. It was a very small molecule, but you have to go to start by the elements more or less. So that was really a long task, and to establish all these things. And at the end, it went out this compound was less potent than other known compounds. So that was really putting a lot of effort into a compound which at the end did not made it out. But then we made some prodrugs for other compounds, and they worked out. At the end, we changed to antagonists because of the more promising class of compounds. And now today I will show you some of the antagonists of this receptor, and in which way these are then going on. Biogenic amine receptors, as you see, biogenic amine receptor will go further with the different structures, and also this is a slide I already showed, so just that we start once more where we ended maybe on Monday or this day, that you get some recognition of things we are going on. And these are all G-protein coupled receptor, and this G-protein coupled receptor, they show the 7 transmembrane helical orientations or structural elements. So for us, they are the magnificent 7, we are always calling it. So then we are going on. I will show you some drug discovery processes in which way we looked for the drug actions, so pharmacodynamics, pharmacokinetics in this way, some compound development in which we made some identification of lead, change of lead, then lead optimization in different structure, selection of drug candidate and so on, and then also some clinical developments which we find very interesting and intriguing really for these arguments, and also in which way we can go on even after clinical trials and go for the different structures. So today I will give you a short introduction in the histamine receptor subtypes, and most of you know the histamine H1 receptor, where allergy is treated, and they have some sleepiness co-effects, so there are also sleep medications for improving your sleep with the histamine H2 receptor antagonist. The H2 receptors, they are involved in the gastroesophageal reflux because they are connected to the acid production in the stomach. And then we have the histamine H4 receptor. This was the most recently one. It's a peripheral receptor which is connected to inflammation and immunomodulation. There were also some clinical candidates already in development, but they did not make it. So they were not better than the other compounds worked so far. And we will focus today on the histamine H3 receptor. It's one of the latest G-protein coupled receptor which was detected by classical pharmacology, so by finding some compounds and then describing the whole behavior. And this is involved in sleep-wake disorders, cognitive impairment, but also on drug abuse in different pathways. And I must stress that we are working here on histamine, and histamine is the decarboxylated histidine in this way. And then you see here, you have the imidazole moiety, you have the ethylamine change, very small, simple molecule. This is really there. And in fact, what I did in my PhD thesis is just to put here on this position two methyl groups here in this way. And they were really worse than the other structure. What we have to know in this way, that the histamine H3 receptor is mainly localized in the brain. Whereas the H4 receptor is mainly localized in the periphery, the H3 receptor is mainly localized in the CNS, and there has some specific function. It's you see here in red the pathway for different structure in the cortex, in the limbic system, and they are strictly connected to the dopaminergic pathway and also to the serotonergic pathway. So thereby, we know that histamine influences the other neurotransmitters, and since the H3 receptor is a control of the histamine there, then the histamine H3 receptor controls not only the histaminergic pathway, also the dopaminergic and the serotonergic pathway in many ways. And this makes it a little bit more complicated. And to get even more complicated, the histamine H3 receptor is a presynaptic histaminergic receptor, which means presynaptic that here it's at the synaptic control, and it controls not only the release of the newly formed histamine, but it also controls the synthesis of the histamine by histamine decarboxylase structure. So then thereby it controls the way. And thereby, if you have an antagonist, it inhibits the negative feedback mechanism and thereby makes some stimulation for the histaminergic pathway. Yeah? So the inhibition is a stimulation. We are going here for the different pathway, and then you can increase your postsynaptic pathway with the H1 or H2 receptor pathway. So there are different possibilities. But you can also have the histamine H3 receptor on the GABAergic, dopaminergic, or serotonergic, acetylcholinergic, and norepinephrine pathway, and so on, so on. So it's a presynaptic receptor on histaminergic neurons and a postsynaptic on non-histaminergic neurons, but not on all of them, but on some of them. And there they have a specific CNS distribution, so more in the cortex and so on. So there are different possibilities in which way you're going on. And to make here the pharmacology really a little bit more complex, then even with the dopaminergic pathway, we do have a formation where we have H3 receptor and mostly D3, some D2 hetero co-formation, where they stick together and influence their signaling, by the way. This was not really known when we started this project, but during the time it was detected that there are some heterodimerization, and this heterodimerization has large influence on the different structures, and thereby the H3 receptor is also influencing mostly dopaminergic, but also glutamatergic and other pathways in this way. And then you see there are some, it inhibits negative function, it increases cognitive function, and has very low effect on the locomotor activity in the different ways. Yeah? That's connected to its specific localization in this way, and I will come back later to this localization because it's important then, because the brain is a lipophilic area, it contains a lot of sphingolipid and other lipid areas, and thereby our compound needs some lipophilicity to go into the correct brain region. And if you then change some physicochemical properties, then you will also change the distribution within the brain. Cannot detect it always, but there are some general effects, and the heterodimerization on different things we are going on for the different pathways in this way. And then, hopefully you remember the structure of histamine, where we have the imidazole and the alkyl chain and then the basic amine. And then we took this as a starting point because nothing was known of the antagonist when we started there. So the receptor was just detected, and then we cooperated with the inventor or with the pharmacologist who has detected this receptor system, and they approached us, and then they asked, can you give us some compounds that are selective only for this receptor? We had no idea, we had no structure, we just had some rough screening models in which way this could be done, and thereby we took the histamine basic molecule, and then to the histamine basic molecule with alkyl chain, we added some longer chain lengths, we changed the other structural elements, but we maintained the imidazole alkyl structure, and thereby we went from agonist to antagonist behavior for the different structure. So we maintained the affinity, but we changed the efficacy of the compound. So by the imidazole alkyl chain, it was a good binder for the different structure, but then with the other structure, it blocked the receptor instead of increasing their activity in this way. And you see some of this compound were very simple. Thiopyramid, these FOB compound. This naming FOB comes from my starting point, where it is Free University of Berlin. So thereby it's this coding from Free University of Berlin. And some of these compounds we developed there got some IUPAC name or UFA names. So like proxifan or ciproxifan, they were renamed after by clear naming in the different areas. And then you can see here, these compounds were active in vitro with below 10 nanomolar inactivity, a good starting point, but getting more lipophilic, we increased the structures, and this compound even in the sub-nanomolar concentration range active with in vitro activity in this way. And that way we had really not big other screening system, and there we went very fast into animal screenings, and there were some mouse model in which our partners then detected the metabolite of histamine, and since we are increasing the histaminergic pathway, we increase the formation also of the metabolite of histamine, and thereby this increase of telemethylistamine, the metabolite, was measured in the brain after per-oral application. And then you see that our compounds, some of these compounds were highly active with per-oral application, and even in the below 1 mg per kilogram in the mice experiment. But we had some problems with proxifan, and this was the example I showed you at the very first beginning. This proxifan behaved not as an antagonist, but as a neutral antagonist, and there it changed not really the behavior at the end. So that was the reason why later on we could detect that this has a very poor in vivo activity because of the neutral antagonist behavior and not the inverse antagonist behavior for the different structures in this way. I hope you remember that the receptor could be in an active and an inactive structure, and then we modify this and we have a basic constitutive activity where this structure concerned. Nevertheless, the ciproxifan, named for the cyclopropyl moiety here and this structure, in fact, this was a compound I prepared only by accident because I wanted to prepare some haloperidol variations, and then I made very strong reaction conditions. And with haloperidol, you have a para-fluorophenyl moiety, and you have the carbonyl moiety, and then have the carbonyl connectivity is a butyrophenol moiety, which has a butyl keto compound, and then I took the chloride to make some alkylation procedures. And then it turned out that the butyrophenone moiety made a cyclization procedure here, and that the fluoro was substituted by the alcohol that I gave. And thereby I got this compound more or less by accident. So I had it in my flask, but it was not the compound I expected. I recognized by different NMR studies and so on that it does not really reflect the expected data. And it took me then really 1 month to go into all of the different detail what I really had there, because it was my first beginning in my PhD studies, and I learned also, and thereby I made by accident then one compound which was later on one of the reference ligands in this way. But I must also show that I did not get the expected, I got the unexpected, but I solved the unexpected. And I did not throw it away because it was not the thing I had. But we tested these compounds, and then we got this compound in this way. And you see it was very efficient in vivo testing. And then we take here the so-called 5-choice task, which is an attention test in the way. With the 5-choice task, the mice is here in a chamber, and then there is some light. And with the light, then they press some button, and then they get some food. So it's a learning behavior and reaction behavior where you can test the attention of the mice. And then you can see here, if you make your stimuli, your light, for half a second, then they are fully correct in all of these behavior with or without the different structures and so on. But then if we give some, or I must say, these mice are not normal mice. They have cognitive impairment by some pre-treatment. Yeah? So they have a very mild cognitive impairment. So their attention is a little bit less than normal mice. But when you give a 0.5 second stimulus, then you see they have about 80%, more than 80% response rate, so they are quite fine. But when you have only a quarter of a second, 0.25 second, then they miss this very often. So their attention is less in this. And when we then give our compound, the ciproxifan, then you see that we're not fully restoring this, but we are getting recovery into this way. So that gives us a possibility in which way then they get a higher attention or getting less cognitive impairment. I must say, when I explain this to different students, then some students say, oh, I take this for my exam preparation because then I get more attention and get smarter. I must tell you that's not the case, because you're just going to a normal level. So only if you have some impairment, then it helps to get less impairment into this way, but not really the compound getting into smarter affinity in this way. Unfortunately, I think. Yeah. Also for us, unfortunate, because otherwise it would be a blockbuster maybe. So nevertheless. And I showed you that these compounds had also activity on sleep-wake behavior. So we tested this compound also for sleep-wake behavior on cats. Yeah? And if you have a cat at home or are familiar with cats, you know that cats are strolling around, sometimes sleeping, sometimes awake, and so on. And you see here, this is the normal way the cats are going. It's a hypnogram where different sleep phases are here. And you see here, the basic state here, basic line, is a wake state. Then we have the light slow wave, the deep slow wave, and we have here the paradoxical sleep, where you have a lot of dreaming like this in this way. So there are different sleeping phases, and you see you do not get directly into the paradoxical, into the dreaming sleep phase. You have to go from one step to another, and then going on, and then this is the normal phase for the cat in this way. And then when we give ciproxifan, you can see here a dose dependency of the different structure from 0.15 mg per kilogram to 2 mg per kilogram. You see that the wake behavior gets a little bit longer in the way. At the end, you have once more the normal behavior with a higher concentration, higher dosage, longer wakefulness, higher dosing, longer wakefulness, and with 2 mg per kilogram, higher wakefulness for more than 3 to 4 hours for the time. And what is also important is not only that they are getting more alerted, higher wakefulness, that when the dosing gets off, then they return to normal sleep behavior. So they do not have any more paradoxical sleep and so on. They turn on to the normal sleep behavior, so we maintain the attention for a certain time and then go back for the normal behavior for the different structures in this way. So that's quite useful where we are going here, and you can clearly see it increases the wakefulness, and thereby it modifies also the sleeping behavior, but only that you're getting more sleeping behavior because you were more alerted during daytime, during taking the drug in this way. And then we tested this compound also for drug profiling in certain ways, so it was quite useful, and we see almost no affinity to other histamine receptor subtypes. We tested them at different species for the structure, always in the nanomolar or subnanomolar concentration range. We have on muscarnergic, adrenergic, serotonergic pathway, and even on the cytochrome P450, we also have almost no interaction in this way, and thereby you can see it's a very pure, nice drug, and only acting on the histamine A3 receptor. And you see, we tested here on rat, on guinea pig, and also on some other screening models, and it was also already highly potent in the different ways. And these were tested in mice, guinea pig, and rat for the different things. But you can see that it's a highly potent, highly selective compound, although it's a very small compound in this way. So we fulfill all this drug criteria: small compound, lipophilicity, stability for metabolism, and so on. But you may see also here one problem we had with this compound. Can you see one problem here? You see with the guinea pig, yeah, we have about 1 log unit lower affinity for guinea pig. And our main target is not rat, not mice, not guinea pig. Our main target is human target. And we did not have the human A3 receptor because it was not detected there by the genomic project in this way. So we just had some mice model, and we did not have really any human models available. So our whole optimization were there on this different rodent models in this way. And there we already saw that they had a good or a very high species relevance. So the species relevance of this receptor from rat to mice was more or less the same, but to guinea pig it was 1 log unit less in this way. And unfortunately to human, this ciprofloxacin is even less potent at human than guinea pig, and thereby we had a very good compound to treat attention deficit with mice, but not really for humans in this way. So our main failure here was that we did not introduce the human screening system, the human in vitro screening system at a very early stage, but I must say it was not available, so it was not detectable in this way, but we failed here in this compound. And thereby the ciprofloxacin now is a very good chemical probe, very good described, with a very high affinity for all animal screening models. Yeah, you see excellent probe, but not really as a good drug for the structure. And thereby you see the difference from a chemical probe, from a good pharmacological tool, to a real drug for the difference. So when you make some in vitro screening with mice and rats and so on, the ciprofloxacin is an excellent compound, but not really for human development in this way. And then we went back for the different structures. So here you see some physiological parameters, because I do not want to go only on our own development, but you see from species to species, you have different parameters. And at the end, you go, for most cases, you go from mice to rat to some other species to pig to monkey, and then you go to human. Yeah? You have to use at least 2 or 3 different species before you can go to human trials. And then you can see that with all of the different species, you have some specialities in this way. Yeah? And in fact, one of our products with the agonistic field, they had some problems with the mice eye, And then we had really to see in which way this problem could be overcome, or what is the reason for this problem. And we could not really detect this problem, the reason for this problem, but that does not allow us to go to human, because we could not exclude that this would not happen with humans. So if you have some serious problems there, the safety issue for human application is really there, and thereby you must be aware that there are different possibilities for metabolism, for distribution, for absorption, and so on in the different species with all of the different areas. But you see here that here the structure of the ciproxifan related structure with the acetophenone element in this way, and then we went here for some other compounds which were also quite useful. We eliminated the carbonyl moiety, but then once more we maintained this high activity for species relevance. And then one of the major breakthroughs was the exchange of the imidazole to a piperidine. So we exchanged the aromatic nitrogen-containing heterocycle to a non-aromatic nitrogen-containing heterocycle, which is very simple from the, if you look on this, so it's just an exchange in this, and this makes even easier synthesis routes and so on, faster synthesis route, faster structure-activity relationship. But I must say, it took us 3 generations of PhD students to find this. Yeah? So we did not exchange the aromatic basic structure to a non-aromatic basic structure. We exchanged this aromat by numerous other 5-membered aromatic heterocycles to 6-membered heterocycles, to different substitution patterns of different heterocycles, different aromatic moieties. So our main purpose really was there on the aromatic structure, and then maintaining this with different positioning, different maintaining, and so on. But all of them really failed. I mean, and I must say, none of these aromatic structure showed any affinity for the H3 receptor. And then you must see, when you work 3 years on this and you're not successful, and then you put another PhD student on this same structure, it's really risky to do something like this, because so many non-active compounds are really shown, which is important to show some structure-activity relationship, because they are very strict to the aromatic imidazole moiety, but even a slight methylation here leads to a drastic reduction of 2 lock units in this way. So very small variation leads to drastic reduction, and larger variation to inactive compounds. And then to come to this piperidine moiety was then the great breakthrough for the different structure. Yeah? So, but you see, it was a long-term bioaesthetic replacement, which was non-obvious for the different structure. But that also gives us lots of possibilities, because we patented this imidazole containing compound, and then the non-obvious non-imidazole compound were also patented in the new application for this way, because they were totally new for the different ways. And then we had this direct binding, and we had some longer chain lengths here with the propyl-oxy-propyl chain lengths. And this compound here with the chloro substituent later on was called pitolisant, and it is very high in vivo activity and also very high in vitro activity. So that was really the compound we looked for at the very later stage for the different structures. But you must also be aware that with the piperidine moiety, these compounds were much more easily to synthesize, and we got a very large structure-activity relationship in this way. And I must say, even the fluoro compound here was a slightly more potent compound. Not statistically significant, but slightly more potent. But why did we choose the chloro compound here instead of the fluoro compound? Why didn't we choose the best compound for further development? Fluoro even has a slightly lower molecular weight in this way, structure, but from lipophilicity it was one thing, but for clinical development it was also one important point, that here we use as a starting material the parachlorophenyl propanol moiety. So that was easily synthesized, and you can commercially buy this parachlorophenyl propanol in a good amount in this way. The fluorophenyl was not commercially available or only commercially available in a small amount. And when we then want to go into clinical development, we want to make kilogram of this and not only milligram. And thereby, it is also important that you guarantee that your synthesis procedure will stay the same for the whole clinical development process, and that you work on the same compound with the same preparation procedure to fulfill NIH criteria and things like this. Yeah? So thereby, it was a very good decision to stay with the power chloro compound, instead choosing the other compound, which were almost the same, but hardly accessible by synthesis, and then you have to prepare the fluoro compound by its own, and so on, which would then get more and more complicated for the different structures. So the different aspects, synthesis is also an important aspect for getting compounds, even if it's a very simple compound for the different way. Nevertheless, at the end we could show that from this ciproxifan starting compound, we stayed with the imidazole compound in the old structure, and then we could add here some elements in this way. With the piperidine moiety, we just need the heterocontaining heterocycle, the aliphatic one. We stayed with the linker, we have different connecting moieties, and then we can add here different pharmacophores, maintaining the affinity of the antagonist compounds. So we increased very much the robustness of our pharmacophore element, and that gave us the possibility to add many different things here at the different structures, made multiple targeting compounds, made fluorophore ligands, and so on, and so on, and so on. So that was a long stay, and that was the reason why I was able to stay for more than 20 years on this compound class, because here we could add so many other compounds to add so many other properties and so on, and the more we understand, the better we can do it. Yeah? And I just added here this Lego particles because we can make it as a Lego brick. We maintain our starting point, and then by Lego brick we add the different structures into the way, by different moieties and so on. But you will see this afternoon where we use these different structures. Yeah? And then you see here that we get the pitolizant main structure here, and then added here different heterocycles, like the pyrrolidine, piperidine, with or without chloro compound, or the 7-membered ring system. And in blue, you see, in dark blue, you see the human histamine H3 receptor, and in the lighter blue, the guinea pig. And you see that these compounds even have a slightly higher affinity for the human receptor than for rodents in this way. So thereby, they are more advantaged in this way, and that makes sense. But it is also important then when you make this comparison to different species, because your previous studies for the metabolic pathway for the dosing comes from the rodent trials. So if you want to make the translation from mouse to man, you must take into account that there are maybe differences in the binding properties, that there are differences in the metabolic pathway. Yeah? In most cases, rodents are much more faster metabolizing than humans. Yeah? They are much more robust than we. They can take almost everything. Yeah? And they have as a thumb rule, 5 times faster metabolism rate than we have in this way. So that's then calculating your dosing when you get first time into humans into this way, but you can see there is no affinity for the other histamine receptor subtypes detected at that time, and that gives us good possibility. But then we had a good ligand for a human receptor, for the human histamine H3 receptor. We know that it had some influence on cognitive impairment. We know that it had some influence on sleep-wake behavior. So there were different physiological or pathophysiological relevance, cognition, wakefulness, memory process, slide on the locomotor activity, slide on food activity, on food intake, on craving behavior for drug abuse and so on. But what disease would you want? So we have, at that time, we had a good compound in our hand, but we do not know the disease, the application, the therapeutic application. Yeah? So that was then in which we go for the different purposes in the way. And for cognition and wakefulness, narcolepsy is an orphan disease which is quite often used. Or increased sleep behavior is also connected to the daytime sleepiness with Parkinson patient under their normal dopaminergic dosing in this way. Yeah? And there are other sleep-wake disorders, obstructive sleep apnea. The attention is difficult in attention deficit hyperactivity disorders, and so on. So there are a lot of potential different clinical indications, therapeutical indications, which could be really performed in this way. But sleep-wake behavior was really the most prominent one in this way. And that was the reason we are using this sleep-wake behavior on mice this time. This time with pitolisant. And you see once more, here's the paradoxical sleep, the normal sleep-wake behavior, and the wake behavior as a baseline in this way. And this is the normal structure we are going here. And for narcolepsy, there were already a drug known, modafinil, and modafinil is a drug which increases the adrenergic and serotonergic pathway, and it's a drug of abuse potential because of this increased attention connected to modafinil, but is the mostly used, the gold standard in narcolepsy. Yeah? And you see here, modafinil makes a large increase of the wakefulness for a long time, and then in a dosage of 64 mg per kilogram with per-oral dosing. And then in comparison, pitolisant in 20 milligram, you see makes almost the same wakefulness into the way by the histaminergic pathway in this way. And when we give modafinil and and pitolisant together, then you can see that they are additive in this way because the histaminergic pathway and the adrenergic pathway are totally different pathways, and then you had an additive effect on this way. The main point here is also that we did not use normal mice for the narcolepsy, but we used the narcolepsy model in which these mice were orexin depleted in this way. And orexin is the neurotransmitter connected to sleep-wake behavior, and now later on be shown to be causally connected to narcolepsy, to this sleep-wake behavior in this going around for the different pathways. Getting the information on the sleep-wake behavior, you now know that this works with narcolepsy, but you do not know the disease narcolepsy. Do we? This should be connected to some commenting system.
His dachshund, Rusty, suffers from narcolepsy, a condition that causes him to suddenly fall asleep. This dachshund, Rusty, suffers from narcolepsy, a condition that causes him to suddenly fall asleep when he's trying to do other things. Little is known about the cause of narcolepsy, except that it can be inherited. It affects humans and animals alike.
This dog really has a narcoleptic disease, and you see he suddenly falls asleep, and this fall asleep is connected to a loss of the muscle tone. And you can easily see that this is really problematic when you're driving a car or when you're standing somewhere, that this narcolepsy is really a strong problem. And I do not want to show any examples for human you can easily find, but then you laugh on the humans, which is not really the case here. So thereby, this narcolepsy is an orphan disease, not so often, and thereby then we can go here for the different structures in this way. So pitolisant was then tested for narcolepsy. You see, we made many other studies. We made molecular modeling with these and with other structures. But then we went into clinical trials with this compound on narcolepsy, and there were some market authorization in 2016 as an orphan drug for narcolepsy. This compound was quite successful because it does not cause any drug abuse behavior. Instead, even it fighted the drug abuse behavior in the different aspects. So it is much more accessible, much more useful for treatment of narcolepsy instead of modafinil or other increasing drugs, which are then under control in the States or even elsewhere in this way. So that was quite useful in this way. And then you see that when you get some clinical data, some clinical trials and so on, I will show you a little bit more on this in the coming elements. But there were even other clinical indication, even photo-induced epilepsy, obstructive sleep apnea, excessive daytime sleepiness in Parkinson patients and so on. So there were different clinical trials on different therapeutic indications. Yeah? And narcolepsy make it out for the first time, and that was also one reason, because narcolepsy is an orphan disease, and with orphan disease you can get an accelerated approval rate, because then this is an orphan disease when not many drugs are available, and with this you can go for a niche into the pharmaceutical development. And I must also say that is also a very nice possibility for the academics to not go for the main players or not having competitors with the main players, that you try to fight Pfizer or Novartis or whatever. Yeah? So Bioprojet here is a small company only interested in new medical entities. They are preparing different compounds, but with a new approach, new medical concept in this way, and thereby this was the very best partner, and they did all of the clinical trials and all of the other things in this way. And I must confess that here, Jean-Charles Schwartz, who is the owner of the, or one of the owner of this company, he was really the driving force getting this compound into market, into the different structural elements. Yeah? But you can see here that clinical trials here is a concept for the clinical trials, in which way you have here the trial for a 5 to 6 week treatment time, and that you have some withdrawal of any psychostimulants before starting this of about 2 weeks, and then you start with the different compounds, you start with different dosing, and you start in comparison to placebo, and you start in comparison to the gold standard. Because at the end you have to show that your compound is better or at least as accessible than the gold standard. Otherwise it's not really going into market really with any success. And then you have paid a lot of money into the development line, but then the agency said, where is the progress? Where is the benefit of this compound, what we did not have already before? Yeah? And then you see there were different dosing tested in this way, and with pitolisant it was necessary to have a slightly increasing doses at the end, and that was the most successful way, and this is compared to placebo, compared to modafinil in a different way. And I must say, this is not the work of the medicine chemist. Yeah? So, but it is useful to know in which way this is really done, the clinical trials, and in which way you have a readout for the different structures and different structural elements. And then you have different endpoints which has to be characterized at the very first beginning. And then after the clinical trial, you make some deblinding for the structures. And with the deblinding, you see if maybe your placebo was the most active compound in this way, or your gold standard, or your real drug. Because here it is a double-blind study that neither the patient nor the doctor knows what they are giving in this way. So thereby, it's clearly done in the different ways, and then going for the different way. At the end, there were 110 patients screened in this way, about 30 in each group in this way, and then there were some dropouts really in this structure, which is the regular way, and then about 25 to 28 people at the end were tested, which is a really low number for screening in this way. Why such a low number, really? Why it was really even accepted to have such a low number? I mean, it's much cheaper to test 100 patients instead of 1,000 patients, but that's not the rule here. The rule is because we have an orphan disease. It's a very rare disease. It is even very difficult to obtain even this 100 patients into a multicentric trial. So there are different problems when you have orphan diseases, because it's a rare disease, and you must then find 100 patients who are willing to change their medication and to to get into this different structure. So the development line for orphan disease is a little bit different than to other regular drugs, which are much more frequent into the different ways and getting it. But you can see that this was already published in 2013, so this is really quite a long story to go for this. What has been found that placebo was slightly active, whereas pitolisant in green and modafinil in red were almost as active as in both cases. In this case, you can see we were as active as the gold standard, but the gold standard is a controlled drug with some drug abuse potential. So there's really some clear benefit for pitolisant in this way, and it has less disturbance within the sleep behavior for the different structures. So thereby it was clearly shown that the drug abuse potential here is shown by these structures, and you can see here modafinil has especially the larger concentration, very, very high abuse potential, whereas pitolisant has a very low one, and that it even has some anti-cocaine behavior, and modafinil is even more abuse potential than cocaine has. Yeah? So thereby it has a different potential, but the abuse potential to get used to this is quite high. And when you then have a drug which, like pitolisant, which doesn't have anything like this, it's quite useful to get into this way. And thereby it was shown that it has a clear benefit in this way. Why is this important? Because modafinil, the narcolepsy, always or can be often starting in your youth, so in your teenage age, yeah, sometimes even earlier. So you have even to treat teenager with a drug, and you do not want to treat your teenager with a drug which has a higher drug abuse potential than cocaine on a regular basis. So thereby you clearly see that there are some potential into this way. So the main outcome here for narcolepsy is the Epworth Sleepiness Scale, where this sleeping behavior was strongly reduced. This is the non-treated, whereas the dosing of pitolisant in this way. And the sleep scale is also true for Parkinson's disease and obstructive sleep apnea. Yeah? You can see here every time the sleep behavior was improved by improving the daytime alertness, by improving the daytime thing. So this drug is giving, pitolisant is giving during the morning, and then you have higher alertness, higher cognitive improvement in this way, higher wakefulness, and with this you have better sleeping in the night. So that's the main point we are having here with pitolisant before and after treatment. And you see here this was approved for narcolepsy. It's called WAKIX in Europe, and also I think 3 years later in the States, and worldwide we do have approval. And in 2020 or something like this, then it was also approved by the European Drug Agency for obstructive sleep apnea by the clinical trials, and then it's treated for the name Osavade for treating this thing. So we have the same drug, both are WAKIX and Osavade are pitolisant, and you see here with 18 mg in the same dosage, but under different naming. Can you estimate why we have not transferred WAKIX into another disease really for this structural element? For why were there two therapeutic indications, and for each therapeutic indication you have a separate drug which goes into market with the same compound? You see, the drug is really with the naming of the different drugs. One point is the behavior of the patient. When you have a drug which is for obstructive sleep apnea, then the people want to take this for obstructive sleep apnea, and they do not want to take this for narcolepsy. So the confidence for the patients is much higher when you have a drug which is only there for a certain instance. So we call it compliance. Compliance is much better when you have this drug. But here, compliance is not the main problem here, because with pitolisant for narcolepsy, we have an orphan disease. And obstructive sleep apnea is not as often as narcolepsy. And especially in Europe, when you sell a certain amount of your drug, then you're losing your status as an orphan disease. Because then you're selling a lot of drug, and this drug is then not really regulated only for orphan disease. And then the regulation officials say, okay, then you get out of the orphan disease status, and then get out of this. So thereby, it is also from a pharmaceutical industry development for getting some revenue payback money in this way, really a good strategy to go into these elements, to have two drugs with the same compound instead of only one drug. Yeah? And I must also say that in the United States, the orphan drug status is similar regulated like in Europe, not exactly the same, but similar regulations. And there, when you develop an orphan drug, then during your patent time, there are no other competitor allowed to make the same drug or make the same mechanism of action with another drug as the same. So because you have developed this orphan drug, which is a rare seller also, then you maintain your priority for the duration of the patent. So there's also some possibility in which way it is really useful for the pharmaceutical industry to get there into orphan drugs and to maintain the orphan drug status then for a while. So that must be aware that it's really possible to go for the different structures in this way. So I do have different possibilities, and here you see pitolisant on Parkinson's disease and different dosing on Parkinson's disease and freezing behavior. But you see the Parkinson's disease really was not really as effective as the others, so it's not really useful. We were not accessible for an additional drug, for an additional indication in Parkinson's disease, although the sleeping behavior improved, but not generally the Parkinson's disease by itself. But we get connected, but not really successful in the different ways. And you will get no drugs with side effects. Yeah? The more effective your drug, the more effective also your side effects are in this way. And here you can see insomnia is one of the main side effects, which is really clear when you make a drug against sleeping behavior, that insomnia must be a side effect in this way. So that's what you want more or less in this way. But that's also important. When you take the drug during daytime, you want to sleep in the night. And we have had some competitors who made some similar experiments with their own development, and they had even some more potent compounds, but with a much longer duration and so on. But then these compounds were keeping alertness, keeping wakefulness during 20 hours or more. So the people could not sleep really in this way. And this pitolisant we choose, this has a half-life of about 8 hours. So when you take it in the morning, then you can go to sleep in the evening. You maintain your regular sleeping behavior. Yeah? Maybe it was luck, maybe it was the genius of Jean-Charles Schwartz and all the co-workers and so on, but nevertheless, this compound really has a good half-life, good affinity, and good structural strength. Yeah? You can see here that there are some dizziness, sometimes some appetite things, or quite often some headache. Yeah? So when you directly start with pitolisant as a large dosage, then you get a headache which slowly disappears. But when you slowly increase your dosage, when you make some slow increase in the input of the structure, then you get a void of this headache, and then the body gets better controlled in this way. So thereby that's really important. And also you must take all psychostimulants, get out of the body before you start this treatment in this way. So there's also some learning we have had, because some failure, some dropouts where the people did not stop with modafinil. They claimed to stop with modafinil, but blood tests really showed that they did not stop, and then they take pitolisant in addition, and then they get headache because of some overdosing of both anti-narcoleptic drugs in this way. So thereby, they went into the different pathways in this way. So clinical trials were done on narcolepsy, sleep-wake behavior. Both of them were really successful within the years. We also made other trials, and other companies made some trials on Parkinson or Alzheimer or attention deficit disorders in this way, but none of them were really successful in this way. Nevertheless, we thought that we have a really good compound in our hand which decreases the sleepiness, which increases the wakefulness, which had some slight influence on drug abuse, which had some slight influence on lowering food intake and so on, and thereby we were looking for some other diseases which this compound could also be obtained in this way. What we wanted to do, at least what is trial, make some drug repurposing, like the company has done with with the obstructive sleep apnea, where they had some other additional indication, some repurposing of the same drug, the drug used for another indication, yeah? Then it was done here for them. So with the increased knowledge for the pharmacological profile in humans, we were looking for other diseases with a huge medical need, and then where we are looking for this drug repurposing in this way. So drug repurposing is the general way in which you can use an original drug and can take it for another indication. Yeah? This is an easy formation. You just have to go to phase 3 clinical trials. You can leave out all other trials because it is just for the patient, and then you just have to show that it's useful for the patient into this way, if it's taken as it is in this way. So thereby the known target, known disease, but then you have some experimental disease where you have mostly the side effects of the drug, and taking this side effects. There's also the H1 receptor blockers. When you have an H1 receptor blocker, you take the second generation for allergy treatment, because this is against the H1 receptor activation you have with the allergy, and then the second generation do not cross the blood-brain barrier. It has no effect on the brain, but it stops the histaminergic effect in the peripheral system. The old H1 receptor blockers, they pass the blood-brain barrier. And by blocking the H1 receptor, they're inducing sleep. And these old compounds are nowadays only used as sleep therapeutics. They're very rarely used. They still have this anti-allergic potential, but they are mostly used as sleep inducers for different diseases. Also kind of repurposing of the drugs for another indication in the different drugs. So thereby, this gives us and repurposing was quite often used for COVID, because we were in desperate need for some drugs in a short time. Yeah? So there were really different possibilities in which you go for this, and thereby I have indicated here some structure, and the remdesivir was already explained yesterday that this compound was successful, whereas many others have not been successful into this way, and also some anti-inflammatory compound in this way. Yeah? So repurposing gives a fast way for new detection, for new indication into this way. Yeah? And by that way, I would also mention that some people suggested hydrogen peroxide into this way as a disinfectant for perioral application. Yeah? The person is now once more President of the United States, yeah? But don't do everything what a non-informed person really do in this way. So that's really, you're not getting COVID, but you're not living anymore. So that's really not recommendable for this way. And here you see the rare disease policy in this way. So rare disease is about less than 6 persons per 1,000. In Europe is less than 5 per 1,000, and in Japan is less than 4 per 10,000 people in this way. So thereby different regulations, different things, very similar but not the same. Yeah? And there are also the regulation for getting into this are really a little bit different. Rare diseases are really rare, so only very few people have them, but we have more than 8,000 different rare diseases, so people having rare disease are really frequent. Yeah? So that must be aware that rare diseases are rare, but many people have rare diseases in this way. So that's really with the whole thing here in this way. And here you can see this is the common or very common diseases, and then you have here your border for rare diseases, and then you have rare orphan, ultra orphan, or hyper orphan with a different structural element in this way. And you can see here that we will go now from orphan to ultra orphan in one of the diseases, and we have different diseases. Because we detected this disease which is also connected to higher sleepiness, to higher daytime sleepiness, and this disease is called Prader-Willi syndrome. It's called, it is connected to hyperphagia, to intellectual disability, so cognitive impairment, to hypertonia, and in many factors really connected to different diseases in this way. This is the typical outcome of a child with Prader-Willi syndrome, and it's a rare genetic disease, or ultra-rare genetic disease. And then we would like, in which way we could connect this genetic disease, this Prader-Willi syndrome, to the histamine H3 receptor. In which way we can connect this genetic predisposition connecting to a G protein coupled receptor. Yeah? So I will just explain a little bit more on the things. So there are maternal uniparental dysomny, where some genes are getting lost or getting deleted in the different pathways, and so it can be inherited because of this chromosomal region. There were 3 different possibilities with the paternal descendation, maternal, or imprinting defects in this way, but nevertheless, the effects are almost the same. It's 1 in 10,000 or 1 in 15,000. So it's a rare orphan disease, even in this way, where we're going in this way. So even more rare than narcolepsy in this way. Even less patients are here in this way. And they have hypergonadism, hypertonia, cognitive impairment, hyperphagia, and very challenging behavior on food intake. Challenging behavior means that the parents had to close the refrigerator. Close the refrigerator by a lock, really. And no food sent anywhere in this way, because they are so craving for food, they're taking anything in this way. And they need a very strict timetable, and you have to stick to the strict timetable. If you change it, then they get into full chaos into their whole life in this way, and they're disturbing into different ways. So thereby, this really is a difficult way when you have such children, and the whole family is affected by the children because of this food behavior, of this other behavior into this way. So this Prader-Willi syndrome, characteristic, short stature, narrow face, the almond-shaped eyes, so physical appearance with hypogonadism directly connect, but you also have a lot of neurological problems where they have temper tantrums, where you have sleeping problems, cognitive problems, and some obsessive behavior in this way. And you see here already with the neurological problems, sleep problems, cognitive behavior, obsessive behavior, this is already connected to what we have seen with the pharmacological profile of the H3 receptor antagonist in this way, for getting in this way. And thereby, here I have listed once more the effects, the typical effects for Prader-Willi syndrome children. So, and then when we go here for pitolisant, we can see hypergonadism and hypertonia could not be changed in this way. So we do not change the genetic profiling, we do not change the epigenetic and so on, so that could not be done. But we can improve the sleepiness, we can improve the attention, we can improve wakefulness, and we can improve the cognitive impairment. Maybe we can improve some antipsychotic behavior, maybe we can improve the obesity, the food craving behavior into this way. And this observation was the starting point that we get into contact with some children with Prader-Willi syndrome. So there are some connectivity in this way, and then this pitolisant could be a good way to test into the different ways. This pitolisant was also in Prader-Willi syndrome mice, where this genetic depletion was induced, and we could see that pitolisant could strongly increase, normalize the behavior on the body temperature, which is the measurement into this mice. So we got into animal studies for a model for Prader-Willi syndrome, and then saw that even successful with an animal model. And then there was a big step that we got into a patient-controlled clinical trial. Because we were asking Bioprojet, our partner with pitolisant, can you do some clinical trials on Prader-Willi syndrome? And their answer was strictly no. We were just successful with this compound in narcolepsy, and we were just successful in obstructive sleep apnea. And these people are multimorbid patients. They have numerous of diseases, numerous of neurological problems. And if we fail here, maybe our drug will be taken out of the market. Not for the reason for narcolepsy, but for some other clinical trials. So we do not go the risk for getting our compound lost from the market, which is fully understandable and understandable. But we got to a patient organization for Prader-Willi syndrome, and this patient organization in the United States, they had two organizations. They had so-called Trend Community. Trend Community is an online thing in which way orphan disease or parents of orphan diseased children report their effects of different drugs and different elements in the way, but thereby got a community of patients with Prader-Willi syndrome. So this trend community, firstly only localized with Prader-Willi syndrome, was one of the things where some patients could be contacted, in which way they can be stressed. And then we got another organization, Chion Foundation. They were founded by some parents with some children with Prader-Willi syndrome. So they were deeply concerned about the development of their children because of the treatment with modafinil and other things, and they were deeply concerned. They were looking for other diseases or other treatment possibilities in this way. So they were very open to get into this. So we got contact with the NIH, we got contact with the patients, we got contact with the parents, we got contact with additional medical doctors who supervise all of the different experiments. And then we started with 3 patients here to get a patient-oriented clinical trials on Prader-Willi syndrome with this drug. These patients you can see here are in the age between 10 and 15 years old, male and female, and with different symptoms for different causes for Prader-Willi syndrome. And in fact, they had only a very moderate body index, because most of them had a body index of 25, 30, sometimes even 40 in this. So these patients were already diet controlled, yeah? But nevertheless, that was the reason why the parents are so eager to learn a little bit more on possible treatment this way. So thereby, and I think a study of this would not be possible in Germany. I'm not sure if it would be possible in India, but it would be possible in the United States, where they have the possibility to have these patient-oriented trials in this way. And in fact, at that time when we made this study, this pitolisant was only available in Europe and not in the United States. So we had to import pitolisant into the United States for these things. So there were different problems in this way, the importing of unknown drugs in the United States, and the allowal for this approval here, and things and so on. So it took us really 2 or 3 years, but in fact it is really fast, and this 2 years was only possible because of the initiation of the parents involved. Yeah? And these parents involved here in the trial, and the parents involved in Trend Community, they all have children with Prader-Willi syndrome, and they were eager to get some new medications. So then we got this trial, and you can see here mental clarity on these things, because mental clarity was a test system, and you see here it was just a subjective measurement. So mental clarity, you do not have any real measurement in this way. So the parents had to calculate in which way the mental clarity of the children get improved or get lowered in this way. And you can see here there is no improvement, and here improvement in this way, or strong improvement in this way, and you see that all three patients got an improvement over the treatment of 50 weeks or even longer into this way. But you see also directly the problems of this way, because here in most of directly you see the improvement with this judgment of the parent in this way. And after some time, they do not see any further improvement in this way. But that is when you have your children, at least my children are small genius, yeah? And they are always excellent in this way. So, and when they get more excellent, you see this. But when they stay excellent, you see, okay, they have a dropout and they're getting normal in this way. And this is the same with the subjective treatment here. After some while, they get used to this behavior in this way, and this is a lower judgment in this way. So that is seen with many of these different trials. And here I have the picture of Kian. He was the first to get this medication in this way, one of the first patients in the way and getting this way. Then processing speed. This is a little bit better here, and here you see some patients got some better improvement, some minor improvement in this way, and it's also that the parents were asked. I got notice that I had improvement in processing speed in this way, but you see here from the starting point, all got a strong increase, yeah, starting point. And then being asleep in the car, because when they get be transported to school or to certain things, they always get asleep. And here, there were no asleep anymore in the car, so they kept wakefulness during daytime when they went to school, and even their grades are getting better into the different ways, and so on and so on. So there's different possibilities. Later on, this study was extended not only with 3 patients, but you see there were more than 14 patients, a different scaling system, and all of them got some improvement to different scaling. And we had really one dropout where the parents avoided to get the washout of modafinil, and this patient got a really bad headache, and he stopped all medication. So that was the dropout which we could really see in the petulant state. And then I would
Since birth, my son Kian has received numerous therapies, followed a special diet with supplements, and received nightly growth hormone shots. He has been periodically monitored by an endocrinologist. Kian was doing very well on this path until about 3 years ago, when at summer camp he was found slumped over and unresponsive to attempts to rouse him. Thankfully, hours later, he regained all functionality and returned from what we thought was a state of unconsciousness. But it turned out Kian could recall everything that had occurred during the episode.
It felt scary because I couldn't really move. My body couldn't really stand up and couldn't really move. And they came in and said, like, Kian, are you okay? Are you okay? Are you okay? I couldn't even talk. I couldn't even explain that I was actually there and listening.
I discovered that my son was probably experiencing a state of immobility. For the past year, all but one individual reported significant improvements in quality of life. Families observed improvements in sleep patterns, daytime alertness, and mental functioning. Many of the children on pitolisant also appeared to experience a normalized relationship with food.
Hey, you made it!
We live in a cycle where It can happen at any given moment, at any given time.
We got to a point where her days got too short because she required the sleep. She'd come home from school and it was a choice. She either fell asleep for an hour or two and then was up for a little while, or she'd make it till about 6.
Justice slept for a good 16 to 18 hours a day.
It was very confusing because I heard people telling me to wake up, but like I didn't know how to wake up. Like I was in my own little world. Like I was saying, like I can't wake up.
It was so bad. Her half days of school became more like an hour and a half of school. Her anxiety sort of manifested in a strange way where she was actually vomiting like 30 times a day.
We were managing him fairly aggressively, the symptoms, because, we were hoping to help him along. But then when the acute incident started happening, it messed up his life.
In school, they really didn't understand what that was and what I needed. So when I did all this thing, they were just like, he's going out of control. What should we do?
I mean, he could no longer do summer camp. He could no longer do some of these things that he was enjoying, theater camp, because he would fall asleep.
We noticed a difference. I actually just had Justice at the endocrinologist just last week, and she said that she cannot believe the difference in Justice. She said she notices such a huge difference. It has been a complete 180.
His sleep started normalizing within days of taking pitolisant. It's like Kian woke up, right? And all of a sudden, not just literally, but figuratively, he woke up.
My body was trying to figure out my new superpowers, so it would wear my body out. Because it was trying to figure out everything that was happening.
I was more awake, I was more energetic, my brain was moving really quickly so I could catch up all the learning that I missed in my early childhood, which was really, really amazing.
I don't have to go to bed at the same schedule, so I can go to dances and stuff and stay up later and watch movies. And at night I would actually fall asleep watching a movie, but now I can actually watch movies.
It was actually kind of hard to cook by myself in the kitchen, because without pitolisant, I was really hungry and I would tend to go in to whatever I was cooking and eat it.
He's a social kid. He really likes to interact. He was much more free, and he was relaxed.
And it was much easier for me to answer questions for my reading. Now I get A's.
I personally am looking at him and going, you know what? You're going to college, and you're going to graduate, and you're going to live independently. You're going to find a partner. I think there's a tangible, defined, independent future.
For families of individuals with PWS who wish to try pitolisant, Xi'an Foundation recommends consulting with a neurologist who is a sleep specialist. As research develops, we will continue to.
So you see that that's really in which we are helping the people, and that's the way we do not forget really when we make chemistry and all of the different things. At the end, the patient must be helped in this aspect this way. And you see, there are some possibilities, and this was just a patient-driven study. Only 10 patients, really, and these are 4 of these patients in this way. And you see that they really benefited from the way. It was a huge way, and it was not really something which was my ordinary work or thing like this. It was totally unordinary going into this patient-oriented clinical trial, getting into all of the contacts with the United States. 2 of them were in Canada, really located. So only with this connection, it was really possible to go for the different structures. But nevertheless, I have to say there are also some problems with the Prader-Willi syndrome, because these people are getting this Prader-Willi syndrome by their birth, and so you have to treat them very early, sometimes even before teenager age or something like this. So there is a difficulty in which way they're going, and when they when the parents see the benefit of the children and then see that they stay at a certain level, then they increase the dosage. And with the increase of dosage, they see some additional effects in this way. And that you see here, that over these weeks, the dosage was highly increased with the different structures, up to 36 milligram with the maximum in this way. And thereby, they get some problems even with the smaller children in this way. So thereby, it's not always the best, and that's the reason why you really have to not take the judgment of the parents, but you have some physicians really included in the way, strongly controlling all of these experiments in the different pathways. So that's really an important way that we cannot go for this into a different way. There are some other elements which are now in clinical development for these things, but nevertheless we published these data, and these data, and with the publication, was then possible that doctors make a prescription off-label for pitolisant as Prader-Willi syndrome treatment. So now worldwide it's possible to make some prescription of this, but still it's an off-label use in this way. So the doctor had to get known and to take the risk of getting into the different structure. So different publication from us in this way. And this Lara Pullen was a lady you have just seen who has founded the Chion Foundation and made many of the contacts. And then she contacted also the US-American distributor of pitolisant, because during this clinical or patient-driven trial, then there were some FDA allowance for pitolisant for narcolepsy, and it was distributed in the United States not by Bioprojet, but it was distributed by Harmony Bioscience. And Harmony Bioscience is a new startup with new medical entities. They are buying them from other companies, and then they distribute them on their own. They are very, very rare own development, but they are looking for different diseases. And they have the license for pitolisant in the United States, and they are declared, based on this trial, we do make a phase 2 clinical trials for pitolisant for Prader-Willi syndrome children in this way. So they really started this clinical trial, and they took advantage of this trend community. They took advantage of the Chion Foundation, because by that way they could add different patients in a short time very effectively. And this is also that the trend community has strongly enhanced their way, and they are now not only focused on Prader-Willi syndrome, they take all rare diseases in their portfolio, and they make some connecting between patients, between drugs, and between patients' reports, and if needed, they make the contacts to pharmaceutical industry to make drug repurposing or any other things in the way to enhance clinical trials, to enhance the property for patients with rare diseases in many different aspects in this way. So that's quite interesting and quite important. And they started really with small, only with Prader-Willi syndrome, with 3 persons, and now I think there are 30 persons and have 80 rare diseases in their portfolio in the So if you look for some, or if you have some people with rare diseases in your community, trend is a good factor even for exchange of the parents, exchange of the patients, what can be done, what can be collected, and so on. So there are different, very nice possibilities in which we are going on this way. And it's also one kind of a side effect we have in this development, that such communities are really growing in this way. So small communities, small factors are really growing into the different ways. So thereby, we are growing. I will tell you later on for the afternoon in which way we can then connect even some other receptor, but here we are just going for some other effects. I'm not sure if I have everything here. I will go, this will be talked later in this way. Nevertheless, I must say that meanwhile, this pitolisant has started phase 3 clinical trials with Harmony Bioscience. So we expect the data next year to get this, and hopefully then pitolisant could also be approved for Prader-Willi syndrome. This example should show you that compound development is a very difficult, a very complex behavior. And we have talking about many different aspects in this way. what gives me really some kind of additional motivation in this aspect is that I get into contact with the different patients where we're doing these things for. So, in fact, I'm very often phoned by some parents in Germany or in Europe who want to get some information, some material about Peter-Lusand, mostly for Prader-Willi syndrome, because they do not have any other contact persons who are really involved and get some information on this. And I must also say there's, with some of these organizations, like the Prader-Willi syndrome, they do have an organization on their own, and they make once a year a meeting. And this meeting is then 3 days science and 2 days community. And community, meeting parents, children meet together, they are making a kind of small family who are always meeting in this way, and they got to exchange what's helped, what's not helping in this way, what's new in this way. So that's really a helpful community to get further details in this way. So with this way, I can show you that Peter-Lusand is a very useful compound where we were rationally developing by long-lasting work into the optimization procedure. This was connected to some luck with half-life and other things in this way, and metabolic stability in this way. This is a very useful drug for narcolepsy, for obstructive sleep apnea, with some positive aspect for anxiety, for cognitive things and so on. So there are many other possibilities in which way they could develop, but they're not successful enough for one therapeutic indication. But when we had the pharmacological profile of this compound, the human pharmacological profile, then we went into further detail. And you see this pharmacological profile was during the clinical trials. So when the data of the clinical trials have been published, then we go into this detail, or when we were informed by Bioprojet what's happening in this way, we already looked for some further indication, because we see in orphan disease is not where you make a blockbuster drug, where you get this large money. And it's better to have a blockbuster drug if you want to earn some money in this way, and that's not done normally with the orphan diseases in this way. Thereby, I hope that you see that medicine chemistry could be much more than just synthesis, could be much more than pharmacy development in this way, and if you can follow your diseases, can follow your structures and so on, then you can easily follow up the fate of the patients in this way. And with this, I would like to close here this way and be open for your questions. Thank you very much. If you have any questions.
Yeah, so the session is open for questions. Yeah.
Good morning, sir. So, sir, when you were telling that sleep wake experiment on that mouse.
The sleep experiment on the mouse.
Yes.
Yeah. Yeah.
So there and at the last, you have shown one point where you did the experiment in the absence of light. Light was off in that experiment.
Yes.
But why not for others? Like if light on, light off, is there some difference? Is there?
Okay, that experiment was that the mice must be trained before to react responsive, to get this responsive. So the mice will be treated, I think, 1 or 2 weeks before, daily, that when they got some light signal, then they will get some food. So they are, when they're taken out of their normal cage, then they are getting in this chamber, and then they know that something will happen, because they are trained on this, and then they got the signal, and with the signal they got the food if they press the thumb correctly. So first is the training session, and then there are the reaction control of this. And during this time, the mice are treated with some cholinergic treatment that they got some cognitive impairment. So they are trained, but they have lower behavior in this area. They are not normal mice. They have a slight cognitive impairment in this way. And then you can check in which way they are alerted or not by this light signal. And if they have a very drastic cognitive impairment, not a mild one, a drastic cognitive impairment. They even change with this 0.5 second signal, but then the brain is too much disturbed, too much, not enough energy, not enough alertness there that they got this. So you must make a disease state with a mouse model that is comparable to elder patients which have a mild cognitive impairment. And that's one of the possibilities. There are many other possibilities which are, can be generated, but that was, I think, to my mind, the most impressive one. And the main problem, we did it also on regular mice, or normal mice, but they react very similar, even if they get pitolisant or not. So this is the control assay, and normal mice do not react on pitolisant with the improvement of cognitive behavior, which shows when you have a certain level of cognitive behavior, which is your normal level, this compound would not bring you above this level. It would just, when you have a lower level, it can restore a little bit of this level. So when you're ill, it can bring you a little bit better on the healthy side, not fully on the healthy side, but a little better on the healthy side. And later on, we could show that it is by improvement of the acetylcholinergic neurons. So the H3 receptor is localized also on acetylcholinergic neurons, and there makes some improvement. And then by that way, you can make some cognitive improvement on the impairment, but not smarter. We were also thinking that it would be maybe a useful drug for military, because the military person could get awake for a longer time in this way, but so far I think they have not been approached. Yeah?
It's just a curiosity, like what is the physical state of this compound? Like what you synthesize in lab, it is like liquid or solid?
It is the hydrochloric salt. So with the piperidine moiety, we have a basic moiety, and then we use the salt of hydrochloric acid there. It's slightly hygroscopic. It has some other compartment, but it's easily dissoluble, and thereby it is, I think, at a log P value of 3.4 to 4.2 or something like this, depending on the calculation method. But that's quite fine, and what is also important that we have a blood-brain ratio. So the ratio between the blood and the brain is more, that we have more in the brain than in the blood. So it accumulates a little bit in the brain, and thereby we have very low peripheral effects, and that is even preferable for the neurological behavior in this way. So by that way, the pharmacokinetics are quite fine. The only thing is deteriorating with this, one obstacle. We have a metabolism, a slight metabolism by 2D6, cytochrome 2D6, and that is an enzyme which is also used by some reuptake inhibitors and so on. So there could be some interaction potential with this. So there are some not fully fine with the metabolism. If you do not take any other drugs, it's fully normal, but there are some slight potential for interaction with some drugs. Otherwise, it is surprisingly clean and everything, yeah? With such a small compound. We are always astonished that this compound has so high selectivity range and can be done in a certain way. And we also discussed some, or will discuss some other possibilities for structural variations.
I know, Professor Stark, it must be extremely satisfying for you to see how your research is bringing about change in the lives of people. And this is a dream of every medicinal chemist, that at least one of his compound can come to the market. So we are very privileged that we are getting to hear to you, and especially participants will be inspired to the research that actually their molecules can go to the market. So I think that participants must also want to know that at which stage you handed over this molecule to drug development companies.
Yeah. in fact, I thank you very much, but the honor for the development is on the whole team, really. So there were Jean-Charles Schwarz from INSERM at that time in Paris, connected to Jean-Marie Lecomte, who was connected to him, and both of them later on they founded Bioprojet. So one person from the academic side, it's the INSERM is something like the Max Planck Institute in Germany, and later on they founded Bioprojet as a private pharmaceutical industry. And Jean-Charles Schwarz was strongly involved in, he was the inventor. He detected the histamine H3 receptor. And my PhD supervisor was for a long time working on H2 receptor blockers, and they met on a meeting, and then they started their cooperation in the H3 receptor field. And when I joined this group as a PhD student, they just had 1 or 2 years in their experience working on this. So that was a very brand new target. We did not knew any association for any disease, and we did not know if we could really develop any compound in this way. Then we had another partner from University College London, Robin Garnelli and his group, and these three groups got together. Robin Garnelli is an inventor of cimetidine, together with Sir James Black, the Nobel laureate on beta blockers and so on. He worked with SmithKline and French, and he had also had long experience in this way for drug. And he went from pharmaceutical industry back to the University College London. So we had a consortium of three groups, the Paris, the London, and the Berlin group at that time, and they worked together. There were two groups in medicine chemistry and one group in pharmacology, biochemistry, and so on. And then there were some developments strongly supported by the European community, by different funds and so on, so there must be some money for this. But the further we get, the easier was it sometimes to get some money in this way. And then when this compound got into a good range, when we had enough compounds with enough drug likeness, then this Bioprojet took over. Jean-Charles Schwarz retired from INSERM and went fully to Bioprojet. So he put all his energy into the compound development and stopped his academic career more or less, or ended his academic career and went to the industry career. And all of the other partners put all their involvement, all of their enthusiastic into the way. And I think that was one of the key points, that all three partners were highly enthusiastic, were running for the same target, were running to getting into the development in this way. And then this compound was handed over, and we were involved in the first drug formulation as a pharmacist, with the toxicological screening, with the phase 1 clinic, in which way the tablets must be formed. But when then the clinical trials were set up in this way, then we were out. Yeah, because that's not anymore that was just with Bioprojet, and we were from time to time informed what progress has been made. But I must say the progress at that time, it had a duration of about 12 to 14 years for the clinical development, which is rather long. So the remaining patent lifetime is rather short, but that's the possibility on rare diseases. To find enough patients for this rare disease takes a long time. To find some expert for this rare disease is difficult. And you saw the person who wrote the paper on sleep diseases, on sleep approach and so on, was one of the experts directly located in France. So it was not only a colleague, but also a friend of Jean-Charles Schwarz. So there are also some personal relationship really necessary for this, and that makes this possible. And I have to say, at least to my personal point of view, from the academic point of view, at least in Germany, I do not know any professor in pharmacy who has 1 or 2 drugs really onto market and is still active in this way. So normally it takes such a long time for the development that you're retired or something like this. There are some possibilities. Yeah? So that's one great advantage. I started very early on this development. Yeah, so with my PhD study. Thank you very much for kind words.
Thank you.
Any other questions from this side?
So always keep enthusiastic on your projects. Be convinced that it's successful.
Good morning, sir.
Yeah?
Sir, day before yesterday you had talked about the chemical probe.
Yes.
And today also you had talked about the chemical probe, which is the ciproxifene.
Yeah.
Would you please elaborate what is the chemical probe and how it could be used in a pharma industry?
Okay. You see, a chemical probe is very good for the characterization of the receptor, or a chemical probe for some pharmacological experiments, for some biochemical experiments. But a chemical probe can be a drug, but it must not be a drug. So when we do some characterization, for example, in mice experiments, then we can easily use ciproxifene, because it's an excellent compound with a good half-life, well characterized and so on. So we do not have to use pitolisant as a ligand and so on, because this is well described, highly selective, commercially available and so on. So that's then used as a chemical probe when you want to test if your screening system maybe is H3 receptor dependent, yeah? Or your compound makes some H3 receptor effect. You test your compound with or without ciproxifene, and if it's the same effect with and without ciproxifene, it's not an H3 receptor effect, because the H3 receptor effect is fully blocked by ciproxifene, and then if you have the same experiment. But if, for example, some additional blocking is then you can perform this experiment. Then the definition of the chemical probe is there. You can have this chemical probe on the receptor level. So receptor binding affinity, binding thermodynamicity, so kinetics, binding kinetics, which is all described with ciproxifene. That is the biochemical or structural level in this way. Then you have the cellular level. The testing on different cell lines with ciproxifene is well described, and the testing with ciproxifene in different animal models, in guinea pig, in mice, in rats, in different rodents, and even in other non-human models are well described. So you can use this compound as a good reference compound for additional trials. But you see from the profile, from the species relevance, it's not useful as a drug, because in human it has lower affinity, and thereby you have to use higher dosages, and then you get a higher probability of some interactions, and thereby it's not a chemical probe for human use, but for many other uses and so on. And thereby you make the definition of a chemical probe, in which way this compound could be used as a reference compound for characterization of this. For example, ciproxifene was used to show that the orexin pathway is important for narcolepsy. So if you block the orexin pathway, the patient got narcolepsy. Yeah? But then it was shown that ciproxifene and other H3 receptors are still effective. So the animal model with the orexin left-out structure has a pathway which does not include the orexin pathway anymore, has clearly a narcoleptic pathway. And then it was shown that the histaminergic pathway is behind the orexin pathway. So if we block if the orexin pathway is blocked, we can still activate the H3 pathway by our H3 receptor antagonist, by the negative coupling in this structural element, and that was really one of the reasons why it's effective. And it was not known when we started. So thereby it's also a nice chemical probe for other structures. You can also use pitolisant, but in most cases when you buy pitolisant, it's more expensive than ciproxifene. And when you design your biochemical experiments, you take a useful, well-described probe, and then take it for your chances. Yeah? Maybe, sorry, may I add one point here? Because I also added this pains, the pan-assay interference compounds, yes? And some of these pains are also described with mini assays, and some are described as useful compounds as chemical probes. And that's really then the difference. These are not useful chemical probes, because they are pan-interacting. Yeah? And that means really the difference in which way this can be differentiated as a useful chemical probe. That's why I explained why we have in Frankfurt this networking for chemicalprobe.org. Yes? And this is really then useful that they are evaluated and described, and not only that some authors say, oh, it's an excellent probe because we have tested them. There are lots of Receptor, redox active compounds and so on, and they have these paint elements, and they are quite often described as useful chemical probes, but they aren't. Yeah, so that's wrongly described in this way.
Hello, sir. I would like to ask if the obsessive compulsive disorder is a rare disease, and is there any experiments going on for that cure?
Yeah. Obsessive compulsive disorder is like a tic syndrome, for example. Yeah, tic disease and so on. And there are also some genetic diseases in which way this can be done. And the H3 receptor are also there in the preclinical phase for trials. So there are different animal trials on their way or already done in the way, describing this tic disorders as a compulsive disorder. But it's also known, for example, that this compulsive disorders like extreme buying, like gambling, like hypersexuality is connected to the dopaminergic intake in Parkinson patients. Yeah, so it's not only restricted to some diseases, it's also some side effects of the Parkinson therapy in this way. So there are different possibilities in which way it is connected in this way, but the effects are moderate, I must say. Yeah, so there are, like with the food intake, they do have some effects in this way, but moderate effects. So they are not, like with the sleep-wake behavior, you see when you take the drug, you keep awake for a longer time. And it's not when you take the drug on the food that you would not take any food craving anymore. It's slowly reduced. So that must be, I'm sure, at the moment I do not believe that it would be successful for these tic disorders or for the others. You must add some additional properties to the compound to be more effective on this. But it's a useful approach, I must say. Yeah? That tic disorder are, they have some genetic background, not only genetics, but they have a genetic background. But it's quite often discussed like the attention deficit hyperactivity disorders, also discussed with the genetic background, but not fully explained. And there, I'm pretty sure that the genetic ordering of this way would be detected more or less by artificial intelligence, because they can broader overview some of these aspects of them. But there are some genetic aspects, but not only.
Hello, sir. Good morning.
Good morning.
What is the effect to take a narcolepsy drug between children and adult person? Because there are a huge difference in brain development and metabolism, etc.
Yeah, you're fully right. And the main difficulty here is that you have to make some trials, some clinical trials, before you can make some children allowance. Yeah? What has been done now, the first trials there were on narcolepsy, were on adults, 18 and older, and mostly older than 22 on most of them. Meanwhile, some further clinical trials have been performed on teenagers, and there are also on children above the age of 6. So from that point of view, the approval also has been done based on these clinical trials, at least in Europe, on children on the age above 6, but below there is off-label use. And I'm not sure, because when you have developing children, they are also developing their enzyme system in this way. And it's hardly overseen in which way this can be fully controlled in every way. And that's extremely difficult to get data on this or to make assumption on this. So when you have a children, newborn, would you treat them with would you treat your own newborn with a drug which has not been tested on children? So you will not get any clinical trials with enough patients on this, even if they had some, only after some time when it's a little bit more grown up and the problems getting more hurting in any way, then I think you will try something like this. At the very first you try it with diet, with some growth hormones and so on. These are allowed on this, so there are other possibilities in which way you can help these children. But for development, there's also one other point. The treatment with pitolisant is only a symptomatic treatment. It's not a causal treatment. We treat only the symptoms. So the disease states. But treating the symptoms means that you can have a better cognitive development. And Kian and Jaden at the moment, they are going to college already. And they do not know any other children with Prader-Willi syndrome that go really to college. Because it's their mental health that they're taking it for a long time, that they make a good development, but also that they have a very strict family, looking on the diets and so on, and looking on everything. And then they go to college. But I must also say, they're not going to college as normal children. They have some housing, they have some people taking care of them, that they are in there. They are still handicapped in a certain way. So they're not exactly normal kids. They are just symptomatic treatment and getting more and more normal, but getting more and more normal social contacts. And as you have seen, when they went to cinema to watch some movies, then she is really watching the movies and not sleeping with the movies. So it's kind of some superpowers. Should we make 5 minutes break and then continue with the other one?
Sir, narcolepsy, how it is different from epilepsy?
Yes. Epilepsy is a disease where you have an overstimulation of certain brain areas. And this overstimulation could be different causes, could be a genetic cause, but could be also damage and many other things. And then you have a breakthrough or breakout by contraction. And there are different outcomes of epilepsy where you have a mild one or a very hard contraction in this way. But this is by an overstimulation of some brain areas in this way. And with narcolepsy, it was really shown 3 or 4 years ago that you have a loss of the orexin receptors or some failures in the orexin receptors. So there's some very specific signaling pathway. And this is caused then by, can be caused by some genetic pathway, which is very rare, but, rare is everything there, but very, very ultra rare. But the rareness here is, in most cases, you have some autoimmune diseases or something like this, where you get some infection in this. So, and thereby you have some, that these neurons are not really functioning in this way. And that's the reason why narcolepsy is not a disease which is happening already by the birth. Normally, quite often it's developing during teenage age or later on. And epilepsy could be by birth, but could also be happening by some very strange behaviors or by accident and so on. Both are brain diseases, but not really connected to each other.
Any other questions for this particular session? Okay, if there is no more question, please join me in thanking him for this wonderful lecture, and I believe it's too much informative enough. Thank you very much. So in between we'll take maybe a 5 minutes break and then we'll rejoin on this particular session. Thank you.